Hello, good afternoon to everyone. It is a pleasure to introduce you Professor Forbes. And well, he's director of the Center of Regenerative Medicine and professor of transplantation and regenerative medicine in Edinburgh University. He's been leading the field of liver progenitor cell for many years, and his lab is working on different regenerative mechanisms in chronic liver diseases mainly. Also, this lab is involved in cell therapy approaches to enhance liver regeneration using macrophages and other cell types, and as well he's working on cholangiocarcinoma as well. So it's a real pleasure to have you here, and thank you very much for accepting the invitation.
Thank you. It's a real honor to be able to speak at Barcelona, and I mean that sincerely. It's been a great pleasure talking this morning. So this is Prometheus. He was punished, and the punishment was to have his liver eating every day. He punished because he let in 5 goals against Liverpool. His liver was eaten every day and regenerated at night. Now, last week I checked and there was 17,977 papers on liver regeneration. It's probably over 18,000 now. And the vast majority of those are looking at normal regeneration. So that is when the normal liver regenerates. So the classical model is to resect 2/3 of a liver in a rat or a mouse, and in 10 days it grows back to the same size. Sometimes we do this in humans, but not very often. In the UK, certainly. Sometimes you may do it more than us, but rarely. We take a piece of liver from the husband or wife, give to the partner or whatever, the family member, and both parts regenerate. And that's the normal liver regenerating. And basically, this occurs through hepatocyte division with no activation of a stem cell compartment. Now, there have been some very interesting papers published on regeneration of hepatocytes, notably a paper published in Cell suggesting that there is SOX9 hybrid hepatocytes emerging from the ductal area in liver. And there is a paper in Nature from Bruce Wang and Roel Nuss suggesting that the central vein hepatocytes hepatocytes immediately adjacent to the central vein are the stem cells and proliferate and regenerate the liver. More recently, there's been a paper published by Jan Torscher's group suggesting that hepatocyte proliferation in the normal liver and in mild injury is spread throughout the liver parenchyma and is not zonal. So essentially, these 3 papers don't exactly correlate with each other. And I think it's an area of future interest, and I think it's an area of ongoing publication. But I'm really not going to focus on that area. I'm going to focus on the situation which we see in the clinic commonly, certainly in the UK, in Edinburgh. And that's typically where you have a severely damaged liver, such as here where there's cirrhosis. And in the UK, and particularly in Scotland, deaths from liver cirrhosis are really rapidly rising, a chronic liver disease. It's the 5th most common cause of death in the UK. Deaths from cirrhosis have doubled in the last few years, and it's a real problem. We do have a treatment, liver transplantation, as you do. We transplant approximately 100 people in Edinburgh every year, but really that's very insufficient for the numbers that require transplant due to the availability of liver. Furthermore, many of those livers are now of questionable quality, and we really create complications. Of course, we save the life of the patient, but then they have ongoing complications from the graft they receive. So really, the question is to try and understand the mechanisms here and to try and do something about it. Now, following severe injury due to any disease, one gets this, of course, scarring. We get an impairment of regeneration that goes along with that, and the liver gets damaged and shrunken and inflamed. Now, one of the things that's been described is that in chronic injury, increasing number of hepatocytes become senescent. so they become P21 or P16 positive, and if you look for markers of cell proliferation, as they fall down as the markers of senescence rise. So those hepatocytes are present, but they're not dividing and they're not contributing to regeneration. And as that situation occurs, one gets a ductular reaction, and this ductular reaction is quite clearly associated with pathological conditions. It's associated with fibrosis, and rapid fibrosis as that. But it's also, some people have suggested, can have a regenerative function in severely damaged livers as well. And this ductular reaction again rises as senescence rises. And this is one of our patients that got transplantation for hepatitis C. This was a number of years ago, was given immunosuppression, they were still viremic, and they got severe injury and developed this severe ductular response and senescence in the hepatocytes. Now, in chronic liver disease in human tissue, for some time people have looked at the liver and seen that these cells are senescent, and within this one has nodules of hepatocytes that are proliferating. And what people see is the spreading out of this ductular reaction, and one can therefore also see intermediate hepatocytes, and these intermediate hepatocytes contain some of the markers of hepatocytes and some of the markers of biliary cells. And therefore the theory has been that when this liver is severely damaged, these regenerative nodules and ductular cells are arising from a sort of stem cell compartment that becomes activated within the liver. Now, because it's human liver tissue, you can't do a thing called lineage tracing where one can trace the origin of the cells, and these are very descriptive studies that people have tried to analyze this. And we really wanted to try and assess functionally whether the ducts have a functional regenerative capacity. And what do I mean by functional? What I mean is, in the sense of the hematologists take stem cells and they transplant them from one mouse. They can take them from the bone marrow and they can transplant them into an irradiated mouse and reconstitute the bone marrow. And of course, that kind of data has led people to develop bone marrow transplantation, and it also indicates that there is a clonal population that can expand and be hematopoietic stem cells. So we really tried to say, could we do the same approach in liver and ask whether there's a functional population. So essentially what we did, and this is published work, but what we did was isolate these duct cells and do clonogenic assays, again as people have done in hematology, to assess whether single cells, if you put cells in one single cell in a dish, can it form a colony. And essentially looking at the colony forming efficiency. And we've screened lots of different types of ductular cells that we could sort on based on their FACS profile. And the cells that were so-called triple positive, that was positive for EpCAM, CD24 and CD133, were more clonogenic than other populations that we were able to find. And we looked at these cells and they could be passaged over multiple passages and retain a phenotype, but the phenotype remained quite biliary and by gene expression different essentially to the hepatocyte population. Now, okay, so we've shown a clonogenicity there, but the question is, can we create a situation, a non-competitive transplant situation, as has been shown for mice using irradiation? So what we did was to utilize a system that we had found had created liver injury, but importantly liver hepatocyte senescence. And we used the AHCRE MDM2 flock system, and this is a very efficient system for deleting MDM2 in hepatocytes. And driven by a CYP promoter, and it creates a situation where P53 accumulates very highly because it's normally inhibited by MDM2, and you get necrosis, but importantly you get senescence. And we asked, and you can see the transaminases go there. And we asked, is this an appropriate model to try and transplant cells and see if they have a selective advantage? So we were able to grow colonies, as I say, clonogenic colonies, and here using a GFP donor, and transplant those into our recipient mice with an MDM2 deletion, and then repeatedly induce deletion in those recipient mice to create a non-competitive regeneration situation. And when we did that, what we were able to show is that very large amounts of the liver parenchyma could be regenerated from these cells. Bearing in mind these are clonogenic populations of cells, so it should really be coming from a single cell. Not only did we find lots of parenchyma regenerated in hepatocytes, there was areas of ducts as well, and these areas look normal liver, completely normal liver, with normal markers for HNF4 alpha, CYP markers, etc. So it looked as though we were creating normal parenchyma. Now in the model where we delete MDM2, we get a very, very rapid development of hepatic fibrosis, and you can see a very marked scarring. But when we transplant into the mice, this fibrosis regresses, and bilirubin goes down, albumin goes up, and the liver function markedly improved. So the transplantation of a clonogenic cell population is really able to repopulate the liver in a widespread sense, but regenerate functionally the structure of the liver and its function. Now, it may be that this is an optimistic pattern compared to the human situation, because here in this situation the transplanted cells, when they expand, are they're not getting any genetic deletion, so they're healthy, whereas the damaged cells are repeatedly being killed and injured, hence the marked fibrosis here. But it indicates the therapeutic potential of this type of approach, potentially. Now in Edinburgh we have a big problem with the quality of our donor livers, and the surgeons will typically complain that they receive a fatty liver to transplant, and that fatty liver really does very poorly when it's been transplanted. The hepatocytes don't do very well, but what these fatty livers are full of are these ductular reactions. They have a very strong ductular response, and so our research program following on from that work is really to try and isolate these cells here in the NLP system, to try and isolate these cells, grow them in colonies, and see if we can develop them as a cell therapy for regenerative medicine. In the same way as islet cell therapy and is a treatment for a form of diabetes or a complication of diabetes, can there be a cell therapy for a specific defect in the biliary system? So as we get increasingly fatty livers, we also get increasingly activated ducts, as I mentioned, but also we can get more of these CD133 positive cells, which are the cells we found to be most clonogenic in the liver. And without going through the details of this, in the fatty liver we have more of an activated stem cell signature in the ducts. And really we've gone to try and do single cell clonogenic assays here, and this is using the Operetta system, which is an automated spinning disc confocal with a robot hand, and putting single cells into these wells. And you can see here the beginning of organoids. You can just see that growing from a single cell organoid, and we can automate that and do screens and based on CD133 positivity or negativity, and see which are most likely to give rise to organoids. And indeed we find the same thing is in the mouse. We find the colony forming ability and the colony and organoid growth is higher in the CD133 positive cells, indicating they may be a good population for us to work with. We've created, based upon this, a mouse model of induced biliary injury and senescence to try and act as a model to transplant these cells into. And again, this is the K19 Cre, which is a biliary Cre, and we express MDM2 in this to try and induce biliary injury and senescence through the same mechanisms that I've mentioned before via P53. And we've made it immunodeficient, and by doing this we can now transplant human cells into this model. And this is ongoing work by John Hallett, who's a student in my laboratory. But essentially, when we transplanted the CD133 cells, the mice have survived. When we give them the induction and do DDC, which is a dietary model of liver and biliary injury. But you can see when they get PBS transplant here, the control, the livers are very damaged, blackened, and really the biliary system in those is very severely damaged. You can see here the widespread necrosis in the controls and the ductular abnormalities compared to the transplant cell population where the livers look very normal. And we can see using markers for human antigens, this is human mitochondria, that we have some areas of ductal regeneration from these ducts. Now it's not complete because the Cre is not complete, but it certainly seems to be a very good way of expanding human cells within the transplanted mouse ductular system, and that's something that we're currently working on. So those are the implications we're trying to develop from the cell transplant situation in ducks. Now, do ducks actually regenerate hepatocytes in vivo is the question. And there's been quite a number of very good mouse lineage tracing papers, and one performed here, which is an excellent paper as well. And essentially, people have used lineage tracing from these biliary driven Cres and created mouse liver injury. And essentially, what they've shown is that these ducks do not regenerate hepatocytes at all, or if they do, it's in extremely low numbers. And the question we had is whether that's due to the mouse model or whether that was due to a biology that was really a very fixed biology. And so also people used labeling lineage tracing of hepatocytes using the AAV8 model, which is very efficient for labeling hepatocytes and does it in a heritable manner to ask if there's liver injury and regeneration, is there a dilution of a population of hepatocytes? In other words, if it was coming from a non-hepatocyte cell, say the ducks, do we get dilution? And essentially they again showed that there was no dilution of this population. But in fact, as I alluded to, in the mouse, when you get injury, you still get very significant ongoing proliferation. These models of dietary injury and recovery or hepatectomy or other models cause lots of damage, but the mouse is very efficient at regenerating its hepatocytes. And so it really, for me, was not modeling the human liver disease. So I asked whether we could try and create a model that would more closely mimic the regenerative potential of human liver. And so to do that, we took an approach using several independent methods, but the first one was really to try and delete beta-1 integrin from hepatocytes using this AAV8 model, which everyone else had used. So we wanted to use the same tools as everyone else had used. And this would either express tomato reporter in wild type or flocks out beta-1 integrin at the same time as labeling. As I say, the labeling is highly efficient. The reason we chose beta-1 integrin is because beta-1 integrin is required for hepatocyte proliferation, and Sabine Werner's group had shown shortly before that if one knocks out beta-1 integrin and does partial hepatectomy, the mice don't survive well and their livers don't regenerate well. There's very little proliferation in the hepatocytes. So that was an obvious thing for us to do. And if we knocked out beta-1 integrin, it's quite an efficient system. Integrin knocks out well, the transaminases rise, there's some liver injury on its own, and you get this so-called ductular reaction, which we've talked about. You can see it spread here, red. K19 is green, but around that is the alpha SMAR red myofibroblasts that accompany it. And what you begin to see now in these livers where beta-1 integrin is knocked out is small patches of unlabeled hepatocytes, suggesting the potential for a non-hepatocyte source of regeneration of these cells. Unlike mice where beta-1 integrin had not been deleted, where really hepatocytes were completely regenerating themselves incontrovertibly. And so we wanted to challenge this by doing injury models on this background. This is a DDC diet where we do injury and recovery, and if we do that combined with this beta-1 integrin knockout, what we find is there's lots of necrosis, and I'll show that in a short time. The proliferation is inhibited initially, and the P21, the senescence is prolonged. So there's a real inhibition of response, and there's more injury. So you can see here these widespread patches of necrosis in the mice where beta-1's knocked out compared to the wild-type mice. And they get a very florid ductular reaction, much more florid than we see in the wild-type mice, that persists and doesn't decrease as quickly as possible. And now what we begin to see in these mice is these significant numbers of negative hepatocytes that are not lineage traced. Here it's between 25% and 20% at 2 weeks, and you know, it's quite a significant number of cells. And we looked at many different models, but this is MCD, a diet of fatty liver, and TAA, which induces a biliary response. And in both of these models, we got basically the same results, negative hepatocytes that weren't lineage traced. And you can see here now what we get is these patches of nodules of cells that are not hepatocyte labeled, suggesting they're coming from a non-hepatocyte source. It could be ducts, but we don't know. It's certainly not coming from hepatocytes, so it's not lineage traced by hepatocytes, and it's around the portal tracts. And we can use a way of mathematically counting each cell and its location compared to the ducts and the veins, using an automated counting system, and we find that these patches are most closely related to the ducts rather than the veins. And these regenerative nodules now, or nodules we think are regenerative, and what we can label them here using pCNA and HNF4 alpha, and we can find that these positive cells are more likely to be proliferative than the surrounding areas. And they look essentially like regenerative nodules you see in human liver disease. By performing whole liver RNA-seq analysis, what we found is the markers of inflammation go up very much, but markers that we've previously shown, such as WNTs, are highly activated, indicating some of the regenerative pathways, notches, etc., are activated that we'd expect in liver repair and regeneration. Now, as you will know, that's all negative lineage tracing, and so really to prove something more definitively, you want to try and use positive lineage tracing. So we use the keratin 19 Cre here to express tomato, and really it's a very quite inefficient system, so it labels at best 30%, but often about 15% of ducts, but it really in the liver does not have any leakiness into hepatocytes. It only labels the ducts, and that's in the normal mouse, and that's in the situation of liver injury and liver regeneration. On its own, it sticks to the ducts. So what we wanted to do is use a non-Cre way to knock down beta-1 integrin, because we couldn't use a Cre system. So we used nanoparticles now, RNA expression in RNAi against beta-1 integrin. And these were made by collaborators in Moscow who supplied these nanoparticles. And what we found is that where beta-1 was knocked down, we now got these patches of positive lineage trace hepatocytes of likely ductal origin, because they contain the tomato Cre, unlike the controls that had a scrambled or a luciferase expressor, where the duct's lineage tracing system did not enter into hepatocytes at all. Now we wanted to try and perform some independent experiments using this, and so we turned to P21 because it's something highly expressed in a human liver disease. And what we did was really just take a gamble that if we overexpress P21 using an AAV system, again a non-Cre system, just overexpressing it, it would have an effect on liver regeneration. And we didn't know if this would work or not. It's certainly an efficient system, but we're very pleased to see it actually worked. So if mice are given control vector or vector with P21 expressor and then given a short injury with carbon tetrachloride and then looking at regeneration, here you can see all the hepatocytes are in proliferation, or many of them, compared to here where there's very little hepatocyte proliferation. There's ducts proliferating, inflammatory cells, but not so many hepatocytes at all. And again, what we showed, which was very pleasing, was to see these red hepatocytes clustered around ducts. Here are the ducts, suggesting that these are the precursor to hepatocytes, but only when hepatocyte proliferation is inhibited, unlike the controls, which are absolutely clean and confirms previous work where people have done this situation. And now we can see here, you can see quite widely distributed areas of traced hepatocytes where there's little ductular reaction, and where presumably there's no differentiation, you get these ducts still persisting. And I think understanding the niche in these different areas would hopefully be very informative to understand what controls differentiation versus what controls proliferation. And when we look at the red tomato hepatocytes, you can see here they look polygonal, they look pretty normal to me. There's my I always show this one, a binucleate one. There are others. There's another one there, but it's the one that my eyes always go to. So they look pretty normal. And the question is, do they behave in a homeostatic manner? So what we wanted to do was try and label duct-derived hepatocytes and then do a partial hepatectomy to see if they could therefore proliferate. And indeed, without going through it in too much detail, what you have to do is label ducts, inhibit hepatocytes with P21, then do an injury, then let it recover, and then do a hepatectomy, and then kill the mice after the hepatectomy. So it's a very prolonged thing to do, and if you do that, you can see hepatocytes proliferating that are labeled. So they are able to enter a proliferative state. So we wanted to do a genetic analysis of this. So from our 3 systems where we have no lineage tracing, where we have P21 derived cells, and where we have cells that are negative patched cells in the beta-1 integrin knockout, we were able to do RNA-seq on those, and essentially the whole genome, using a principal component analysis, the whole genome expression pattern from the duct-derived hepatocytes and the so-called indigenous hepatocytes was very similar and different to the ducts. In the beta-1 integrin system, again, there is some difference here, and I suggest that maybe because of the abnormal niche that persists in this model, but that needs further validation. And this is just what we're able to do is do a clear, if you can actually clear the histology and do a scan through it, and essentially these traced ducts going to hepatocytes very much like leaves on a tree, and you can see the sort of tree formation here. So if you just follow it out, they're all connected and they all branch back to an original branch point. So that's just sort of focusing in on one, turns around, and then comes back again. As you can see, it's all connected really. Okay, so I think I'd like to say that induced hepatocyte senescence models clinical liver pathology. It triggers a ductular response, and these ductular cells can regenerate hepatocytes, but only if hepatocyte proliferation is inhibited. We therefore have a model to try and understand the molecular controls of this that we can try and target human liver disease with. Okay, so that got us thinking about senescence. I'm not a senescence biologist, just to say that, and I know there are some very good senescence biologists in Barcelona, so I'm slightly wary, but Essentially, we're interested in looking at biliary senescence because we wanted to ask the question whether biliary or epithelial senescence is actually driving the pathology and not just a consequence of injury. And I think lots of people have shown that hepatocyte senescence occurs in chronic injury, but the consequence of it, and is it an actual driver of disease, has really not been looked at very much in the liver. So what we looked at is essentially we looked at cases of primary sclerosing cholangitis, which is very common in the UK, and primary biliary cirrhosis. Again, these are common in Scotland in the north, where you get lots of biliary disease. And what we found is that there is lots and lots of biliary senescence, which goes up with increasing disease, but is irrespective of the age of the patient, just to say that. The disease patients are actually younger than our normal controls. And you can see here they express, again, markers of senescence on the bile ducts. We wanted to model this, so we used this K19 Cre MDM2 I mentioned earlier to induce P53 high state and senescence in the ducts. And essentially what we then find is that we do get senescence in the ducts on its own, but it aggravates biliary injury. So if we do biliary diet, DDC, we get more fibrosis, and we get less proliferation, and we get more injury. So biliary senescence induced by MDM2 actually amplifies the disease. If we perform a partial hepatectomy in these mice that have had MDM2 deleted from their ducts, we get a terrible regenerative response here, you can see here, compared to how it should be, indicating both that the ductal epithelium is required to be active and proliferative in liver regeneration after hepatectomy. But what we analysed when we saw that, which is very interesting, is proliferation is really turned off in hepatocytes. It's not just turned off in ducts. Proliferation is inhibited in hepatocytes. You can see here, there. And that was quite surprising to us. The mice didn't survive, they did very poorly, and we got lots and lots of senescence, P21 positive cells in hepatocytes, which kind of made us think about, can senescence spread from cell to cell to cell? Not to linger on this slide, but just to say we've looked at one of the signals of this, and the TGF-beta pathway is highly active in these mice, and by gene and other methods we've looked at it. So the TGF-beta pathway is activated. And so we wanted to look at TGF-beta inhibitors to see if this would inhibit the pathology using this model. And indeed, using small molecular inhibitors of TGF-beta, we get less fibrosis. But interestingly, we also get less senescence. And we restore the regeneration, the Ki67, back up to our normal levels, so it allows the ducts to proliferate again, and this regenerative response is really important in controlling the fibrosis, I think. And it could be that senescence in the ducts may be a trigger for fibrosis, but that's unproven. So that's all been about chronic disease really, but what was really surprising is when we started looking at acute liver injury. And this work's been led by Tom Bird, who was originally a PhD student in my lab, but now has his own group at the Beatson in Glasgow, and he's doing very well. And essentially what we found, and in collaboration with other people I'll mention later, including Tanya Roskams, obviously in Leuven, who's a great liver pathologist, what we found is that in acute liver injury in humans, we saw lots and lots of hepatocyte senescence, and this was very unexpected for us. So we get very commonly fulminant liver failure in patients with paracetamol or acetaminophen overdose. And we get lots of necrosis here, you can see the necrosis. But what we found was that lots and lots of senescent hepatocytes around this. So the hepatocytes are there, but if we look, they're P21 positive, P16 positive, and they're not proliferating. So they're not contributing to regeneration. And the more the injury we have, this was from a series of non-A2E patients, subacute liver failure patients from Tanya's group, is the more the injury, the more the senescence, and paradoxically, the less the regenerative response. So that should be going up, and it's not, it's actually going down. So we wanted to look in our animal models to see if we could model it, and then could we develop mechanistic understandings of this. So we used just wild-type mice, giving them carbon tetrachloride or acetaminophen, and indeed we saw basically the same thing as we see in the humans. We see lots of senescent cells adjacent to the areas of necrosis, and these are HNF4 alpha positive hepatocytes. They're P21 positive, but does the P21 actually have a pathological role in this? So we performed this experiment in, or Tom did, I should say, Tom Bird, in P21 knockout mice, and he found that in the P21 knockout mice, what you do is you lose this ring of senescent hepatocytes. So these perinecrotic senescent hepatocytes suddenly go away, but that's not unexpected. But more than that, what you now get is a restoration of proliferation in those hepatocytes. So they then become regenerative, and what we can do again is restore the relationship between injury and regeneration. So more injury, more regeneration, compared to what we were seeing in the wild-type mice, is where regeneration goes very high, more injury, less regeneration. The paradox. So what was interesting then was that we found that by genetically inducing senescence in some cells, what it looked like is that we were transferring senescence to adjacent cells. So one way we can look at this is doing a partial induction of MDM2 in hepatocytes by using a low dose AHCRE or using a low dose of the AAV8 virus and titrating it. And what we found is when we could induce single cells that became senescent, that were senescent and were genetically deleted and high P53, surrounding that became cells that were senescent that became P21 positive but hadn't had the genetic deletion, indicating that the cells next to senescent cells were catching the senescence, if you like, almost like an infection spreading through the cells. We've looked at this in multiple ways, including in cell transplant models, and what we find is in hepatocytes that are transplanted into senescence, the senescence can spread into those transplanted hepatocytes. Oops, sorry, just go back. Again, we looked at the mechanisms of this, and what we found, sorry. We looked at the mechanisms of this, and again what we found was it was TGF-beta driven. So the signaling pathways of TGF-beta are highly activated, and so this gives a question of whether we could target this. So I won't labour that aspect too much, but sufficient to say it was activated. And by giving TGF-beta small molecular inhibitors, what we found, and given paracetamol in high dose, is that we're able to restore survival compared to non-survival in a high dose paracetamol model, and in a lower dose paracetamol model we're able to reduce senescence and restore regeneration. So a molecular inhibitor of TGF-beta now, not genetic but a small molecule inhibitor, is able to restore the relationship between injury and regeneration, which is lost in the situation where we get lots of injury. Obviously this would be a very intriguing way forward in the clinic, and it's something we'd love to try and develop further. So in summary, senescence drives liver pathology as well as being a consequence of liver damage. It's active even in acute tissue injury, which was a surprise to us. TGF-beta is a mediator. It's certainly not the major or all of the mediators, and it can be targeted. So just in the last part, I'd like to talk a little bit more about how we're developing cell therapy approaches. So very finally, what we want to do in cirrhosis, of course, to regenerate this, we have to reduce scarring as well as stimulate regeneration. So just putting in transplanted iPS-derived hepatocytes in cirrhosis, I believe, is going to not work on its own. And we believe there's a niche where the macrophages are very important in controlling this regeneration, and I'd just like to show you some data on this. So what we showed previously is that macrophages are required to program the regenerative response from ducts, and if you delete macrophages using liposomal clodronate, these ducts don't spread out into the parenchyma, they just curl up as tight biliary structures, and they have very high markers of HNF1 beta, the biliary marker, and HNF4 alpha is lost. Unlike here, where macrophages are present, these ductular structures can spread out and HNF1 beta is low, and markers of hepatocyte markers are increased, and we can isolate those cells and look at their genetic profile and basically show the same thing. So macrophages are required for the patterning of the regenerative response in injury. And indeed, they're able to eat the dead material, and when they eat dead material, including dead hepatocytes, it actually helps macrophages to start expressing Wnt ligands. And we've looked at this in regeneration, but we've also looked at this in in cholangiocarcinoma, where it can be pathological. So essentially what we found is that macrophages, we wanted to test them as a cell therapy, and we got a surprising result when we were injecting macrophages into normal mice with no injury whatsoever, because if we injected bone marrow or macrophages into tails of normal mice, they get a ductular response and their livers actually grow bigger for a period of time, for a few weeks, and then they shrink back to normal. And essentially what we were able to show using genetic knockout, this was via tweak FM14. Also, macrophages are required for the remodeling. If you take away an injury, even a cirrhotic liver can begin to remodel. However, the more damage there is, the harder it is for that liver to remodel. If there's mild injury, it remodels well, severe injury, less well. And essentially, macrophages are in the scar form. Scar-associated macrophages are actually there, and they vary in the resolution phase. And John Iredale showed with Jeremy Duffield that deleting those macrophages led to less scar resolution, suggesting that macrophages are important for this. So we wanted to try and develop this as a cell therapy, injecting macrophages into mice, using a mouse model of carbon tetrachloride with a single injection of macrophages, and essentially you get less scarring, more regeneration, and matrix metalloproteinases are increased, and you get a regenerative response. These are not polarized macrophages, these are just differentiated mature macrophages using CSF1 to differentiate them. And they express chemokines, so you get an amplification of the effect by bringing in endogenous cells to amplify this scar resolution and regenerative response. And these livers, the albumin's improved and the function's improved. So we wanted to try and develop this as a clinical therapy, and we are doing this. It's called the MATCH trial. That's me having my monocytes removed using apheresis. I've got billions of them, as you have, so it's not a problem. You take the monocytes out, you can put them in a bag, and then differentiate them into regenerative macrophages. We want to develop this as an autologous tool, so there'll be no immunosuppression, and hopefully no consequences of immunosuppression. And to do this, we had to show that the macrophages you get from healthy volunteers can be differentiated into regenerative macrophages, as we show in the mice. But even cirrhotic patients, we could do the same thing. So although cirrhotic patients have very abnormal monocytes, when you take them into ex vivo, out of their niche, and culture them, they can regain their mature macrophage function ex vivo. And if one looks at the gene expression pattern of healthy volunteers versus cirrhotics, you can see it's pretty closely, there are some differences, but on the whole it's similar. But do they work? in the NOD-SCID mouse, the immunodeficient mouse, where we induce carbon tetrachloride-induced fibrosis and give macrophages, human macrophages, you get less fibrosis. And myofibroblasts go down. The effect is less dramatic as we see in normal mice because they have they don't have obviously an active immune system, so we believe there's less amplification effect. And we've now shown the same with iPS-derived macrophages and with Leslie Forrester, and that's now published. So the trial, we're doing a phase 1 dose escalation study in patients with stable cirrhosis, and essentially we take out the monocytes, differentiate them, reinfuse them, and we look for safety outcomes. And essentially it's been very well tolerated in our patients where we've done a dose escalation and we achieved our aims of maximum dose. I can't really discuss the preclinical data on this at the moment, but it was sufficiently positive to make us want to do the randomized control trial. So we're now in phase 2, performing the randomized control trial. We do this in Edinburgh. I hope you'll come and visit us. Here's the Centre for Regenerative Medicine, where we have a GMP facility, the Scottish Liver Transplant Unit, the hospital, the imaging centre, the clinical trials facility, and the apheresis machine. So everything's on hand to try and do this. As I say, we're now doing repeated infusions of macrophages in our patients with cirrhosis in a randomised control trial to see if they work or not. Now, there's many things that have been shown to be successful in mice that have not translated to humans, unfortunately. I'm sorry to give you the bad news. And in fact, this is one of them, and this is work I did with Phil Newsome, who's in Birmingham, who we did this study, and this was based on work he'd performed in mice showing that CD133 hematopoietic stem cells were antifibrotic and could stimulate regeneration in mouse models of repair, and also lots and lots of cardiological studies suggesting that these cells could be regenerative, and lots of small studies in patients suggesting benefit. Well, unfortunately, we looked at patients with cirrhosis and they had standard care, G-CSF alone or G-CSF and these cells, and I'm afraid we got absolutely no effect whatsoever. In fact, there was a suggestion of potentially more complication in the patients that got G-CSF or G-CSF cells, so that was a negative. So I'm hoping the macrophages will be more positive, but I emphasize the need to do randomized controlled trials to know this. So I'd like to summarize and say that macrophages are involved in regeneration, they have promise in animal models, and a clinical trial is underway. And I'd like to thank my group who've been fantastic in doing this work. Sophia's done the biliary senescence work, Wei and Alex did the ductular work, Wei did the transplant work with Tom Bird. Tom Bird's done the senescent work recently in hepatocytes, and Owen's a collaborator who's helped us with lots and lots of mouse genetics. So thank you very much. MELD 10 to 16 outcomes are basically changing MELD and UCALD, clinical events. Also, we're doing lots of biomarkers, the Nordic biomarkers, ELF. We're scanning them as well. They're getting MRI and MRS. So it's essentially quality of life as well. So everything we possibly can look at, we're looking at. Also, we're looking at the serum markers of everything, including cytokines, inflammatory responses, etc. The MRI MRS will be quite interesting, I think, because we're looking to see if the liver is metabolically different, structurally different in size, shape, fibrosis, etc. So we'll see. No, we're not tracking the macrophages. So tracking the macrophages is possible, but it's quite difficult at the moment. If we use SPEOs, the iron particles, they actually change the phenotype of the macrophages, so we're not keen on that at all. In our animal models, we've looked at multiple different settings, including liver injury. When we inject macrophages via the tail vein or the peripheral vein, they always hone to the liver. Obviously, they go via the lung, but they always end up in the liver and spleen. We can't stop them going there. So they don't last, they're short-term cells. So in mice we find them up to a month maximum. Well, for sure, one thing is severity. So we know that from the paper. The more the severity, the more the likelihood of senescence and failure to regenerate. There's a tipping point, which is paradoxical and unexpected. Beyond that, we have no idea. So we don't know anything about that question. But it's a great question, and it's something we really need to understand. If it's just TGF-beta driven, maybe we've got a therapy or something, but it may be that there's multiple factors. We don't really know that. Okay, thank you. I got that. Okay, so about the fibrosis, I think that's a really excellent point. So can I just comment? The quickest I've ever seen cirrhosis develop is in a patient of mine with fibrosing cholestatic hepatitis. Can you hear me? Can you hear me? Yeah, it's in fibrosing cholestatic hepatitis. This patient had taken 20 years to get cirrhosis with hepatitis C. Post-transplant, he had too much immunosuppression. He got cirrhosis again in 3 months. So there is something about the ductular response that drives fibrosis very, very, very rapidly if it's uncontrolled. There's lots of TGF-beta, there's lots of collagen, there's lots of laminin, which people don't think about in fibrosis. Also, there was lots and lots of hepatocyte senescence, so it could be hepatocyte senescence driving it as well as the biliary reaction. But I completely agree with TGF-beta. In the biliary disease, such as PBC, PSC, it is possible that biliary senescence is driving pathology there. So you're quite right. So we really need to study this now. I think we're kind of at the beginning of this rather than the end of it, but I've always believed that biliary, this ductular fibrosis we see is of a different quality to the carbon tetrachloride or hepatocyte-derived fibrosis, but I'm not a fibrosis expert. So we haven't done those models, but I think that's a really great idea because we know that senescence in hepatocellular carcinoma, they have a horrible linkage, and whether biliary is the same, it could exactly be the same thing. That's a really good suggestion. Yeah, it's a very good question. So I need to say one thing. The P21 overexpressing virus is not actually true senescence. It's inhibiting hepatocyte proliferation, but it's not truly creating the classic senescence secretome we see with the MDM2 knockout. MDM2 knockout, we see classic senescence. So it doesn't just have to be a pure factor. Whatever's in the P21 works very well as well. So it must be something more related to inhibition of hepatocyte proliferation rather than purely just a senescence thing.
Okay, so it's not something that is secret.
I don't think it's just like if we take IL-6 and squidge it on, it's going to work. I think it's something more complex. It's a very good question, and it's one which we haven't studied in any detail, to be completely honest. But let me just say a couple of things. Paracetamol injury often is in the young, who, you know why? There's some disturbance and they take the tablets, okay? So even in young people with normally healthy livers, we know that they were healthy, they get significant senescence. But I wonder whether in non-A to E injury, the injury may not be so specific. I don't know if it's a virus, a toxin, whatever we see. It may be the senescence and lack of regeneration that defines that as a syndrome. Because those patients often don't survive unless they get transplant. If they survive, their livers shrink, and they get kind of a pseudocirrhosis. You must know these patients, but we get lots of them in Scotland for sure. Then your question about
Total reaction in Egypt.
yeah, it's not something I've particularly looked at. So to be completely honest, most of these mice are sort of 8-week-old mice, sort of standard thing. So we're really not doing a good justification to age, but we should do.
Okay, well, if there's no more questions, thank you very much for
Thank you very much.
for coming here.
Thank you.
Thank you.